Do I have to repeat my Spanish now? I will. Me encanta Chile. Mi primer viaje a Chile fue en 2003, de Nueva York a las Islas de Pascua, y este es mi 10º viaje a Chile y el 5º Magallanes. Como piloto de avionetas, he visitado muchos lugares en Chile, desde Arica hasta Castro. El sábado llegué al fin del mundo después de viajar por más de 34 horas, pero al llegar me sentí inmediatamente como en casa. Mañana voy a visitar por primera vez Epithelium y de vuelta en un día contado por el estrecho de Magallanes. Solivaldo, eres un gran anfitrión y espero que mi presentación les agrade y les sea de utilidad. That was my best that I could do. So I'm a Spanish teacher now in Frankfurt since 3 years. He's Mexican, so my accent is a bit Mexican. And we used to do this. I used to say cara a cara, but now it's casa a casa, right? It's all virtual. And I'm happy to be in a Spanish-speaking country and practice a little bit. Okay, I think this is working now. Thank you for your patience. Al fin del mundo. So I've been working the last 2 years, more than half of my time on COVID. I brought my own COVID machine to El Fin del Mundo, this Apple ID now. It's not a PCR machine, but a machine based on amplification of RNA. And I did another test this morning in Questo, in La Hotel Jaguar Loca, and it was negative. Okay, so just so that you know, this morning at 10 o'clock, I have to fill that in every day for the say 19 to say I'm negative. I will try to move this thing here a little bit because it's really in the way now. How do I get rid of this? Look to go. Okay. Can I get rid of it? No? Oh, it will disappear. Yeah. So olfactory dysfunction, el perdido del sentido de olfato. Probably you all know someone who has had this problem, and we don't understand the mechanisms. We as a community, and I'm very interested in contributing to that. So it's about half the people who contract the virus who will have some kind of olfactory dysfunction during the disease. In 90% of the time, it is transient, but in 10% of those 50%, so about 5%, it's persistent, and we don't like to say that it is probably permanent or lifelong, but it's better to say persistent, right? Because if you say to someone you cannot smell anymore for the rest of your life, that's not a nice diagnosis. The patients are a little bit more females than males, and usually on the younger side, 30 to 60. And what about recovery? If it's transient, it's fast, in a few days often, and a few weeks. And if it's persistent, we don't know. We don't know if there will be recovery, potentially lifelong. And many patients recover from the anosmia into a parosmia, which means things smell very differently, usually badly, and sometimes phantosmias. That means they're smelling something, but there is nothing to smell. It's really a mystery why the sense of smell is affected, why it recovers in most cases, and why in some cases not, and why in some cases it leads to parosmia. Now, to sample the olfactory system, to take pruebas is very difficult. It's not like taking a little bit of blood or a biopsy from the skin. And why is that? The olfactory mucosa is anatomically scattered islands. I call it an archipelago, many islands high up in the nose. You cannot distinguish one island of olfactory mucosa from the surrounding respiratory mucosa by color or by texture. Endoscopically, it's not distinguishable. And then microscopically, to make it worse, particularly in adult people, some areas have no neurons. They look like olfactory epithelium. There are basal cells, macular cells, but no mature neurons. So this is really not easy to study in adults, people like us. The olfactory bulb, there are two of them in the brain, in the cranial cavity. Well, you cannot really take a biopsy, it's impossible, because you have to make a hole in the skull and pull out the bulb, so forget about it. That cannot be studied in living patients. That's why we started to work almost 2 years ago on dead patients, so post-mortem cases, where you can take out all these tissues. So the tissues that will be taken out, and I'll tell you in a moment how, are here the respiratory mucosa in the concha inferior, concha media, and the concha superior, that is all respiratory mucosa. Then in the olfactory cleft here in green. Is it coming out okay? Yeah. That's where you find olfactory mucosa, the little islands, right? Not one whole sheet. And then finally here in white, these are the two olfactory bulbs. So the projections are ipsilateral. So right to right and left to left. There is no crossing, no chiasm like in the visual system. So this is the procedure. It's quite a usual procedure. It's post-mortem and it's bedside. So soon after the death of a patient, and we'll explain more in detail exactly how this all went, a team of otorhinolaryngologists, ear, nose, and throat in English, goes to the bed of the patient and removes tissues endoscopically. Extremely quickly. So within 1 hour, 1.5, maximum 2 hours, the tissues have been removed, which means we have very good quality of tissues. These are almost like biopsies, right? But it's not an autopsy. It is something in between. My two main collaborators are in Leuven. That's my hometown. It's also Catholic University. I'm also Catholic, at least born Catholic. And the person who started all this is Laura McCarran. She's an otorhinolaryngologist with endoscopic expertise, and she learned it actually in Barcelona a few years ago. She's fluent in Spanish. And then we have a student together, Michael Kleisters, who's a young medical doctor. He's also fluent in Spanish, by coincidence, and he's the one who has been doing a lot of the work, frontier work there in Leuven. And we call our project Inosmic 19. It's a complicated acronym. I already forgot what it is. Yeah, here. And of course, COVID-19. Manik made a fantastic video of this procedure. It will take about 4 minutes. I will just let it run and talk to you a little bit. So this is in the COVID patients last year in, actually 2020 already in November. Terminate is concha, so concha inferior, media, and superior. This is the olfactory cleft, which is very narrow, 2 to 3 millimeters, and in there is somewhere islands of olfactory tissue. We also do a swab, SOPO, I think you call that, yeah, nasopharyngeal post-mortem to really do prova rapida, a rapid antigen test, and see at the moment of death how much viral load there still was. So this is all endoscopically, and these are then removed and put in immediately in formalin, little container of formalin from Sigma. So inferior, middle, and superior terminates have respiratory mucosa. Some papers claim superior terminate can have olfactory mucosa, but we have never seen that in more than 100 cases. Okay, so this is superior and middle terminate, concha, and then finally the olfactory cleft mucosa. You can see how difficult it would be to remove tissue from a living patient, right? This is really, really difficult to get to anatomically. So through the roof of the nasal cavity, the sharp knife, and you have the fila olfactoria. I will tell you more about this in a moment. These are little olfactory nerve bundles that go from the nasal cavity to the cranial cavity. And these little nerve bundles penetrate the labilla cribriformis, cribriform plate. That's actually a Spanish word, cribra, right? It means a sieve, also in Latin. So it is, the part of the skull here is perforated as little holes, about 20 on each side. And through that go these olfactory nerve bundles, fila olfactoria. So the virus is very close to the brain, right? It's up here in the nose, it causes less of sense of smell, and then it could get, in principle, quickly into the brain through the fila olfactoria. That's still an open question. So normally this transcribiform procedure is made with ultrasound, but because this is COVID patients, it's too dangerous to create aerosols, so they use a hammer and chisel. And this cribriform plate is very thin, by the way. It's not a big piece of bone, you can cut through it very easily. Now you see the dura mater, and then here is one bulb, bulbus olfactorius. It's quite a big structure, 2 or 3 centimeters long in human, very long. And then olfactory tract going to the olfactory cortex. And so Laura is very fast at this. She normally does it in living patients, and then she does it a bit slower, but that's her job, endoscopic endonasal approach. Gyrus rectus is part of the frontal lobe, which we also take samples from, but I won't talk about it today. And at the end, take a little bit of cerebrospinal fluid, very clear fluid, with a syringe to see if there is also viral presence in the CSF. And then give it on the gyrus rectus. Yeah. And here we have an idea of how big all these samples are. So that was it. That was the video that Malik made from one patient in November 2020, COVID case. We also have control cases. So it's an endoscopic endonasal transcribiform approach through the lamina cribrosa, it's also called, through the cribra. And then the samples are put in formalin and sent by FedEx to my lab in Frankfurt, Germany, where we do typically RNA-scope and immunohistochemistry. I will tell you in a moment what RNA-scope is if you don't know about it. A few hours ago, we had another case in Durban. Died in the middle of the night, this person. Case number 95, and right now the FedEx is going to my lab and will arrive there tomorrow, and then we will process it. So that's what this was the last but one case, COVID case number 94. It is Delta. We don't have Omicron yet, and we typically get these two towers of samples from the left and from the right side, and formalin inactivates the virus very quickly. So it's safe to send it by FedEx. RNA-scope, that's an ultra-sensitive method of RNA situ hybridization. I took this video, I think, from YouTube somewhere, just in case you have never heard about it. It's based on very special probes called double Z probes, so Z. They are synthesized by this company that has the RNA-scope technology called ACD or non-biotechnique. We don't know the sequence of these probes. They don't tell us, but they're very good at that. And then you can see where all the specificity comes from. It forms like a big Christmas tree, right? On one RNA molecule. So one RNA molecule will become what they call a dot or a punctum. I prefer the word punctum, right? So we have one punctum, many puncta. So just each time you look at one RNA molecule, that is signal is amplified like crazy. Beautiful methods and almost no background. So the signal to noise ratio is very high. Our first publication came out a few weeks ago in Cell. So this is this Laura van Gerwen. I'll show you a picture in a moment. Marnik, who made the video. And then Mona, who's in my laboratory, the scientist who was leading the work together with London postdoc Cheryo Yu. So this is done. The patients were all recruited from Belgium, from two universities, the one in Leuven, academic hospital. So that's where I'm from. That's where I was born, and I went to medical school. And then a second hospital in Bruges, Brujas, I think in Spanish, a beautiful town. And we have 20% or so of the patients come from there. We have now expanded it to Brussels, Brussels. We have three centers. This kind of study is better done with more than one center, right? Because you never know what specifics you will find in one population or the other. So this is Laura, who came up with this idea. She called me 2 years ago. I will still never forget that, the 2nd of April 2020. Called me on my mobile phone, and we knew each other before, and said, I have an idea, and I will do post-mortem endoscopic procedure in COVID patients. That was April 2020, right? Very, very early days, no vaccines. We didn't really know how it was spread, so there was a bit of a risk for them to do this. And then Mona is on the other side. She's in my lab. She did beautiful images. She spent for this paper, I think, 1,000 hours on the confocal microscope to get the best images and representative ones. So this is the cohort that we published in the Cell paper. There's actually 70, but 2 of them were what we call convalescent. Oops, there's no space there. 70, so 2 were convalescent. They had recovered, went back home, and then a few months later they died of something else. So we don't do this anymore. So basically, we have in our cohort that was published 68 cases. More men than women, that's known in general, right? The mortality is more with men than women, but the olfactory dysfunction is the other way around. It is more women than men, and also an age difference. The median is quite high, 80 years old in our study. And then the number one risk factor for mortality is actually obesity, but it usually goes together with diabetes mellitus, right? Obesity you can control a little bit. You can control your body mass index, but diabetes you cannot really control. So you see here all the risk factors. And we focus now on a very acute phase of infection, more about this in a moment, within death in less than 2 weeks. So that's the paper comes directly from our paper. The patient who died this morning in Belgium in a hospital, he got a diagnosis yesterday. So he died 1 day later. Old person, three vaccinations. So the vaccines don't protect you against mortality, or also not against Omicron. Now I will introduce you first to the samples we have. We have control cases, more than 20 now. So these are patients who also died, but not with or from COVID at the time of death. And we do maybe a 1 in 4 ratio. So for every 4 COVID patients, we have 1 control patient. And we do that all the time throughout the whole project. Right? It's always important in science to have good controls and not just do the controls first and then all the patients. Right? In fact, in one case, one of our control cases had COVID, but no one knew it. This patient had a swab 48 hours before he died, and we took the tissue and said, oh my goodness, there's an infection here. So this is probably the earliest infection ever seen, I think, in the literature, this control case. Not this one on the image, but another one. So the respiratory mucosa, there are 3 parts in it, in the mucosa: respiratory epithelium, then you have the basement membrane, lamina basalis, which is quite thick, and then the lamina propria. And in the lamina propria, you have a lot of glands, and these glands make mucus, snot. Yeah. Is that moco in Spanish? Really a lot of it, right? And that's one way of getting rid of pathogens, of course, to produce a lot of fluid. So these are the 3 parts of the epithelium. In the epithelium, you find these white vacuoles. These are the goblet cells. These are cells that make also mucus, and in the H&E staining, they become just white, translucent. Our artist made a schematic diagram of the main cell types in the respiratory epithelium. And more than that, but these are the main ones. These are the ciliated cells in pink. Then in green are the goblet cells, which would show up white in H&E. Oops, that happens sometimes. I do that. That's a difficult slide, this one. Okay, right? Sorry about that. So we have the ciliated cells, we have the goblet cells, and then at the base of the epithelium are the basal cells. So this epithelium regenerates very, very quickly, fortunately. It makes goblet cells, ciliated cells, and other cell types really, really fast in the order of days when it is destroyed, except when the basal cells are destroyed, of course. And then in red is one example of gland and a duct that goes through the respiratory epithelium, and that's where the mucus goes through. So you have to think about the respiratory epithelium as perforated. There are lots of little holes in there. Each time a duct ends in there. And in fact, the virus can go in there, and as you will see, it sometimes infects the glands as well. The ciliated cells have cilia, right? Sometimes they're called multiciliated, and they're all pointing backwards, and they're moving. If you put some dye in the front of the nose, within 30 minutes, it's at the back of the nasopharynx. And that is one of the main reasons why it's best to take a swab all the way in the back, because that's where the virus concentrates, right? Not in the front of the nose. That is really useless in my view to do a swab. Okay, so now I will show you our typical 4-color images. So we have DAPI to stain the nuclei. You all know about that. We have then 1 or 2 RNA scope probes for the RNA, and that's written italics, the symbol, and then often at the end an antibody, and that is in Roman characters, right? Like here, EPCAM, epithelial cell adhesion molecule. This is MUC5EC, one of the mucins that is in the mucus. So it is here within the goblet cells, and here, just in this section, we had a duct, a gland going through the epithelium, and then secreting basically free mucus. And now is FOXJ1 with RNA-scope. Remember again, if it's italics, it's RNA, it's RNA-scope. If it's straight, it's an antibody. So FOXJ1 is a forecat transcription factor. It is a very good marker for ciliated cells, and in fact, in FOXJ1 deficient ciliated cells, there are no cilia produced. So FOXJ1 is required for ciliogenesis, and that's the main marker we've been using in our work. And these cells, ciliated cells, are the target for SARS-CoV-2, as you will see in a few minutes. Again, FOXJ1, puncta with RNA-scope. TIMPRESS2, the famous TIMPRESS2, an extracellular protease that cleaves the spike protein so that it can fuse the membrane. And then, even more famous is ACE2, ACE2, angiotensin converting enzyme 2. It's very difficult to image because the levels of expression are not high, both at the protein and RNA level. Often it doesn't work. I mean, not just for us in the field. So you have to get a very good sample, a very good technique, and then you can see ACE2 at the protein or the RNA level. So in the respiratory mucosa and epithelium, you find a lot of ACE2. It is all here at the surface apical. So all the virus has to do is diffuse through the mucus. It's got the receptor there, and it's got the protease there, and it's inside cells. Right? That's why it's so good at replicating here in our nose. Everything is ready. From another control case, olfactory mucosa, again, we have the olfactory epithelium, we have the lamina propria, and then the basement membrane, which is quite thin compared to the respiratory mucosa. And in the lamina propria, we don't have so many glands anymore, a little bit, but mostly axon bundles that go to the fila olfactoria, that make fila olfactoria and then go into the bulbus olfactorius. In the olfactory epithelium are basically three cell types. We have the basal cells, which are the stem cells. We have the olfactory sensory neurons expressing the odor receptor genes, and then we have the glia-like cells, sustentacular cells. And the Swiss cells are the target for SARS-CoV-2. I can tell you that already now, and I will show you several images. So again, the olfactory mucosa, it's actually the epithelium I'm showing here. I'm simplifying now. Three cell types: basal cell stem cells, lobose and horizontal basal cells, producing olfactory sensory neurons throughout life, including in adults, and then sustentacular cells, glia-like cells. The olfactory neurons don't have cilia, stereocilia, but they have cilia, they don't move. And in these cilia, that's where you find odor receptor proteins and the signal transduction cascade. So when the odorant diffuses through the mucus, it binds on the cilia and then activates cyclic AMP and so on and so on. It happens very apically. All right, this is a COVID case. We have no longer a virus in it, so we use it as a control case. This is a marker for sustentacular cells, GPX3, glutathione peroxidase 3. These sustentacular cells have lots and lots of enzymes. They are catabolic. They have to remove, detoxify odorants and other things coming in the nose, and GPX3 is one of them. Very good marker. Keratin 5/6 antibody labels the basal cells, the stem cells, and then here in black is the basement basal lamina. Remember, not a stick as in the respiratory mucosa. And then finally here olfactory neurons we can label with NO2. That's a chloride channel at the end of the signal transduction pathway. So the odorant binds to the odor receptor protein, membrane G protein copper receptor, activates a G protein and a cyclase. It makes AC3. It opens cyclonucleotide gated channel. And then finally, at the very end, you have a chloride channel, ANO2. That's the final step in the pathway. And that's a very good marker for mature olfactory neurons. Cytokeratin 8 we use a lot as a nice antibody marker for sustentacular cells. OMP is olfactory marker protein. It is a marker for olfactory neurons with no known function. And the puncta for a gene expressed in olfactory neurons typically form more like a cherry, right? Little thing. And then I wouldn't know how to say this in Spanish. A little stalk to it that is a little bit bent. That is a very typical pattern of the puncta that are in an olfactory neuron, and we can recognize actually with the shape easily the cell type. Now, this is one of the many odor receptor genes in the human genome. They were discovered by Richard Axel's lab in '91 in Cell. I did my postdoc with Richard Axel shortly after that. They both got a Nobel Prize in Medicine in 2004 for this discovery. In the human genome, there are 389 odor receptor genes in the mouse. The latest count is 1,144, intact, with an intact open reading frame. And this picture, what I'm showing you, is the first picture ever published of expression of an odor receptor in human epithelium in 30 years. I found it myself hard to believe, looked at the literature very carefully, and it shows you how little developed our field is. It's related to the fact that it's difficult to take tissues, but up until this picture in our paper in Cell, no one has shown expression of an odor receptor gene by RNA-scoped hybridization or immunohistochemistry. We took this one on purpose, OR5A1. It has a ligand called beta-ionone. The smell of beta-ionone, it's used very often in perfumes and foods. And there is actually polymorphism in human beings that determines a threshold of detection. This is a very special case. There's a nice paper about this published years ago. Depending on what allele you have, you can smell beta-ionone at higher or lower concentrations. That's why we took this one as an example. Took 3, you probably all have used or know about neuronal tubulin, also with microphone factory neurons, keratin 8 for testicular cells. And then again, ACE2, very difficult to show. And you find little bit here and there. Not as much in the respiratory epithelium, where it's almost everywhere, but in the olfactory epithelium, it is more, probably a bit more patchy. Also, TIM-CRISPR is expressed. So in principle, the olfactory epithelium is also a target for the virus. There are 7 known human coronaviruses. This is the one we are now all struggling with. We had the other one, 2002, 2003, with only 8,000 or 9,000 cases, right? The first virus. Then in red here is the one in the Middle East, coming from camels, the very virulent virus, caused a lot of mortality. And then, quite interestingly, these are 4 coronaviruses that you and I have been infected with many, many times. You don't get immunity to coronaviruses, by the way. It should be hinted that the first one also, you won't get lifelong immunity. These 4 coronaviruses in yellow here cause the common cold very easily, very frequently. And the reason why I'm showing that is one of them I find quite interesting. That is called NL63. I think that comes from the Netherlands, number 63. It was discovered only in 2004. And why do I find it interesting? That it also uses ACE2 as a receptor. So of these 7 human coronaviruses I showed you, 3 uses the ACE2 receptor, SARS-CoV-2, SARS-CoV, and this one, but only SARS-CoV-2 causes anosmia. So in my field in 2020, many people thought they could explain anosmia by defining the cell type that expresses ACE2. It's not enough because this one doesn't cause anosmia and it also uses the same receptor. Okay, I hope you understand me. One shouldn't think too reductionistically about it. It's a very big virus, SARS-CoV-2, very large RNA virus, single-strand positive sense. Positive sense means it can be translated into protein. Influenza is also single-strand, but negative sense it needs to be replicated first in positive sense and then it can make protein. It's got a bunch of open reading frames. ORF1ab is a very big one. It's 2/3 of the genome. And ORF1ab is then translated in 16 NSPs. You don't find many papers about it. There's no antibodies against it. I found nothing, no antibody, no probe we could use in our studies. So there's 16 of them, and it's one big polyprotein, and then it's cut proteolytically in different ones. There are a few of interest. This one here, non-structured protein 12 RNA-dependent RNA polymerase, which is the target for molnupiravir, right? One of the antiviral drugs. There's also proofreading enzyme in there, by the way. And then the one I find very interesting is NSP1, non-structured protein 1. It is a very powerful viral protein that shuts down initiation of translation of host messages. It blocks the whole of the ribosome through which the mRNA comes, so there's no more translation of host mRNAs. And in addition, it causes the decay of the existing host mRNA, cytosolic, through an RNase pathway. So this molecule, this protein NSP1 really takes over the cell. All the RNA is degraded and no more protein is made. In the end, you end up with a ghost of a cell, right? And you will see some pictures of that. Unfortunately, it also decreases the expression level of the markers we use to define the cell types. That's with the characteristic shape and the position in the epithelium and so on, we can still figure out what is what. So no antibodies are known. This diagrams you find, this is in a review in 2020, annual review of microbiology. It's a very complicated diagrams, but I didn't want to make my own one because it would probably not be right. So you have the docking and the ACE2, then TMPRSS2 is the major pathway for spike protein. Then inside the cell, the RNA is liberated. It's positive sense, so it can be translated immediately. And then these non-structured proteins set up a very complex machinery called RTC, replication transcription complex, NSP2 to 6D, whereas NSP1, remember, that's the one that's shutting down everything in the host cell. Okay? Now I have to show you that briefly because there's one point I want to make. This virus, this positive sense single strand, it is transcribed into negative strand genomic RNA, which is an intermediate for replication, and that gives rise to, again, positive strand, which goes into the viral particles, right? But then to make it more complicated, there are small subgenomic RNAs made from the 3' ends, and those are then transcribed again in positive strand, and those give rise to spike nucleocapsid E, what is E again now? Forgot about that one, and membrane protein, the 4 structural proteins. RNase code has probes for these negative sense strands. They're not called antisense, they're called sense probes. And if the cell has RNA for the negative sense strands, then it's thought to be undergoing actively replication, right? So we're using those probes to distinguish between a cell that has just viral RNA and one that is producing, is having an active replication transcription complex at the time of the analysis. It's a bit complicated, I know. It's an important point, I think, from our work. So we have the spike protein, E from envelope and membrane. They're all membrane associated. And the only one that's not membrane associated is the nucleocapsid protein that floats around the cell, it fills the whole cell, and it looks for single-strand positive RNA and then grabs it, and it wraps it into a virion for viral particle. And I'm mentioning that because nucleocapsid is filling the whole cell, it's the best viral marker we have. Many papers in the literature use spike antibodies, but it's only membrane associated, it's not always so clear. Whereas nucleocapsid, it fills the whole cell, and you will see in a moment, immediately see what cell type it is. Okay, so only one full-length positive strand typically is assembled into a virion, and then it buds by exocytosis. It's not really lytic, doesn't really kill the cell as far as I understand, at least not initially, but the virus just been produced by exocytosis, right? By a host cell that has become a ghost. It's only producing the virus. The swimmer plot, that's what I call it in medicine. And time 0 is the time the swab was taken that led to the diagnosis, and then each patient swims and then dies, right? That's what they call a swimmer plot in medicine. We had of these 70 cases, remember again, 2 were convalescent, so 68. 30 of them were what we call informative. They had still viral RNA and viral nucleocapsid protein at the time of death. All these patients, except one, died within 16 days after diagnosis. One of them lived much longer, to 29, and that one was an immunosuppressed patient, the recipient of a solid organ transplant in ICU, is producing virus and virus and virus, right? But that's an atypical case. So basically, 16 days after diagnosis, we can still find the virus in these tissues. After that, we don't. The ones in red, the 9 cases, those are the ones which had booked out for the sense probes, detecting negative sense RNA molecules. So these 9 cases had active ongoing viral replication at the time of death, and they died in less than 9 days after the diagnosis. We start time 0 at the time the swab was taken. That led to the PCR diagnosis of COVID. Other papers in literature use the onset of symptoms, duration of illness, but that's very difficult to determine in many cases. When exactly did the symptoms start? Particularly if it's an older patient in a nursing home and so on. So we use time 0 is the time the swab was taken that led to the diagnosis. So now we get finally into several COVID pictures, and then my talk is over. This is one of our best cases, COVID case number 7. This person died late October 2020, 4 days after a negative swab. So he or she had several swabs for one particular reason, negative, positive, and died 2 days later. So we could really say the timing of the infection was a few days before the time of death. Nuclear capsid protein fills cells in the respiratory mucosa, epithelium, almost every cell. Then remember again, the sense probe detects negative sense RNA of the coronavirus. So these cells are now replicating the virus at this time, the time of death. The puncta for the sense probes had a very typical location there around the nucleus, perinuclear, and the puncta for the other viral probes are everywhere in the cell, as you'll see in a moment. Here is a magnification. Right? Every cell is infected here, they're ciliated cells, you will see this in a moment. They're almost all undergoing replication, and the cell is full of nuclear capsid protein. Same patient, nuclear capsid in a different color. This is another sense probe specific for N against perinuclear puncta. And then another sense probe, so we combine 2 sense probes in 2 colors to really show ongoing viral replication convincingly. Another case, COVID number 27, we just numbered that, started at 1 and now we're at 95 this morning. This is a probe against not the negative sense strand, but normal virus, or 1AB, and you can see that the puncta are everywhere now, right? And not anymore around the nucleus, that's only the negative sense strand, but the normal viral genome doesn't show puncta everywhere in the cell. And this is then the sense probe, perinuclear. Again, almost every cell is infected. COVID case number 51, FOXG1, member, microfaciliated cells, probe for membrane. Again, in italics, it's a probe, and clearly it's a FOXG1 positive cells are the target for the virus. Again, combined with replicating RNA. So that's our typical 4-color analysis, right? DAPI, 2 RNase co-probes, and then actually in this case 3 RNase co-probes or an antibody at the end. That was the respiratory mucosa. So the main target cell type are the ciliated cells. In some cases, 4 cases out of the 30, we found that the glands in the lamina propria are infected, and in 3 of the 4, it was only the glands of the lamina propria and not the ciliated cells, which is a very unexpected finding. Now we're in the olfactory mucosa, another case that was very informative, COVID case number 8. This is a non-infected region. Didn't mention that yet, but the infection is patchy, which is not a complication to analyze it. It's not infecting the whole mucosa, with a patch here, with a patch there. Sometimes a single cell, right? Maybe it forms the epicenter of a patch of infection. So here is an uninfected patch. We use a new marker here. I haven't shown you that yet. UGT2A1, that is UDP glucuronidase transferase A1, which glucuronidase using UDP lipids in cystinoduclar cells. Now we have an infected patch, and you find some ghosts here, right? Some dark cells, and these are the ones that are highly infected, full of RNA with a probe for nuclear capsid. Another field of view, UG2A1, a marker for cystinoduclar cells, Ermin, that is a marker for microvilli on top of the cystinoduclar cells, and here again you find these 4 or 5 infected cells. To me, the shape of a cystinoduclar cell is not like a Prosecco glass, right? It's sour maybe here. It is a very typical shape. It has a kind of a broad apical side, then you have the nucleus, then a very thin top, and then it sits on top of the basal cells. It really grabs them. Very different than the cherry-like structure that olfactory neurons represent, right, in this type of microscopy. Here you see the extent of the infection. This epithelium is almost entirely infected. 1 cell after the other, the patient died 4 days after diagnosis, except for 1 little patch that is negative for the virus and still has cytokeratin, right? And here you can see it already. That's my favorite picture here. This is a single isolated cystinoduclar cell undergoing active application at this time, right? It's got a sense probe for negative sense RNA. The GPX3 puncta are absent here because that cell is already losing all its host mRNAs very quickly in a few hours, but it's still recognizable as a sustentacular cell because of the morphology, thanks to nuclear capsid, which stains the whole cell, right? The viral protein is everywhere. And now I will finally show you negative evidence. We cannot really show in science that something is not the case, but we have never seen infection of olfactory neurons, and that was somewhat controversial in our field for our paper. Can olfactory neurons be infected, yes or no? If they can, we have an explanation for anosmia, and right, the virus can get to the brain quickly. So that was a very important point for us to focus on, but the evidence is negative. We have not been able to show infection, so most likely it is not a target cell type of olfactory neurons. It's only the sustentacular cells. So here a probe, pool of 4 probes was used for 4 different hormone receptor genes, and you find a few cells here, a few cells. You will see the next few slides very easily, I think, clearly, that, so these are the cherries. Remember again, olfactory neurons, there's a pool of 4 probes for hormone receptor genes. These are the sense puncta for replicating RNA, and they're in the apical layer where the sustentacular cells are. So it's mutually exclusive. The viral RNA is in the apical layer, the sustentacular cells are, and not where olfactory neurons are. Same case again, that's really our best case. Another pool of 4, massive infection. Right? This is why it is showing you such an extreme case, because if you cannot even see puncta here for the virus in olfactory neurons, then we probably not going to see that in other cases. Right? Because this is really a massive infection and very early. Now, Omicron, to end by that. Omicron is 7-8 weeks old. Friday, 26th of November, the name was given, was baptized by the WHO. It's a small O, Omicron, and then the big O is Omega. That would be the last one, probably. And I was very struck by an interview that came out the next day with a physician in Pretoria, South Africa, who basically more or less discovered the new variant. And she said that she saw unusual syndrome of COVID, all the symptoms except one. The sense of smell was not lost. Right? So we went like, aha, that would be the La Jave del Misterio, right? If we can now get Omicron cases and we cannot show infection of sustentacular cells, it would be a very simple explanation, right? A variant that infects sustentacular cells can cause anosmia, and this one not. But it's very early now. This is only 6 or 7 weeks. And we don't have an Omicron case yet. Maybe the one from today was Omicron, I will know on Wednesday. We do PCR. One PCR reaction to differentiate between Delta and Omicron. So that's the way we're thinking about it now. This new variant could provide the clue to the mystery. This is a graphical abstract of our paper in cells. SARS-CoV-2 comes in the nose. It's got a lot of target cells. The respiratory mucosa is 95% of the nasal mucosa. There are lots and lots and lots of ciliated cells, huge number. They express ACE2 and TMPRSS2. They're all infected quite easily. The olfactory epithelium, these little patches, it's only about 5% in the nasal mucosa. Sustentacular cells express ACE2 and TMPRSS2 and are the target cell type, and not the olfactory neurons, we believe. Then I didn't have the time to show that, but in a few cases, we're working on that now actively. The leptomeninges around the olfactory bulb have viral RNA, but no ongoing replication. It's in our paper, but I don't have time today to discuss that. So leptomeninges is Tia mater and arachnoidea. When I was writing our paper, when we were writing our paper in for Cell in May and June, I was totally struck by this study, which is now in present Nature Genetics. You probably know the 23andMe project in the United States. And in this study, they had 70,000 people, probants, and they asked them self-reporting, have you had a positive SARS-CoV-2 test, yes or no, and have you had a loss of sense of smell or taste, right? And they found one loss, just one, by GWAS, just one, and that is these 2 genes, right? And one of them is expressed in sustentacular, in fact, both of them in sustentacular cells. So it's very consistent with sustentacular cells, which express these genes, to be the onset of this whole chain of events that leads to loss of sense of smell. perfectly consistent with our findings, but doesn't prove it, of course. So this is the Innosvic, part of the Innosvic 19 team. I was in books a bit too early. Glad it worked quite well. Yeah. So this is Laura, who initiated the project, the otorhinolaryngologist. This is Mona. Laura pushed and Mona pulled. In between, we were all in there. Then Marnik is the main person in Leuven who has been dealing with the patients and the sample collection. He is a medical doctor. Another medical doctor, a colleague of him, is Wout Bakker, who did also many cases. This is a pathologist in residence. They're all flamencos, like me, Flemish. The flamencos outside, it's the same word. Soy flamenco, pero no un flamenco. Flemish, but not a flamingo. And then this is a new student we got a few months ago, Sumit Choi from Korea, who works in my lab. So that was it. The study is ongoing. System-tacular cells, that's where it all starts. Fortunately, they are regenerated quickly. That's maybe why the anosmia is usually transiently, right? That's some easy hypothesis. We don't think olfactory neurons are infected, and in some cases, I didn't have the time today, the leptomeninges around the olfactory bone have viral RNA, and that may come from the cerebrospinal fluid. We don't know yet. And again, this is our favorite picture. Typical shape of the system-tacular cell, devoid of GPX3 puncta, because MSP1 has been causing decay of the host mRNAs and actively undergoing replication at the time this picture was taken, an isolated cell. Okay, what's the time now? Not so bad. Thank you. Just give it a microphone. Yeah.
Thank you very much for your very nice talk. I'm one of the persons that suffered this anosmia. I lost completely my sense of smell, and still I'm not recovering totally. And my question is, what's going on in this case? How you can explain that? What happened after? I see that you can identify precisely the site of infection, locations, cells involved, but what's going on after? Why, or what's going on in the people that are affected? The virus is still there. The cells are damaged. I want.
Can I have the microphone?
Thank you.
Yes, so the virus is not there anymore. We're not the only ones, but I think we probably have the best evidence. The virus is cleared very quickly from the nasal respiratory mucosa within, let's say, 2 weeks, I would say. Still, those people died, those patients died, right? But they don't die because their nose is infected, obviously, the lungs and all the tissues, but the virus is gone. There is one paper, quite a stupid paper, I would say, that claims that some patients with persistent anosmia still have virus in their nose. Maybe you have seen that paper, but it's really, really bad paper. And they make strong conclusions that that's important for public health because anosmics could maybe still infectious and so on. Just forget about that paper. It was in Science Translational Medicine. There's really no evidence for viral RNA or viral protein or anything after about 2 weeks. But the damage has been done. In most cases it recovers, in other cases it doesn't. In our cases, we had 6 with infection of testicular cells, 6 cases. It's a very acute phase. I mean, they died too quickly in a way, right? There was no time for them to become anosmic and then recover. And that poses a challenge for us, because at some point we have to address people like you. Laura is thinking intensively of submitting an ethical protocol to do it anyway in living patients under general anesthesia volunteers, right? Not easy to organize that, and then take a very selected biopsies and hope you will get olfactory mucosa and what can be learned then. It could be a quantitative issue, right? If 90% of your olfactory mucosa is damaged, maybe it's too much and it can never recover. I think it's 80% it can recover. So that's another issue, the quantitation. In the short run, I mean, the whole field has problems with that, studying that, not just us. In the short run, what you can do is some patients who have persistent anosmia after COVID, who have to undergo nasal surgery or something else, to then take tissues from them. But there's no literature on this. Lots of hypotheses and explanations, but there's no hard data, no molecular biology, no histology on people with persistent anosmia. It remains a mystery. But you can understand maybe intuitively if there is too much damage done to the testicular cells, maybe they cannot regenerate, right? That would be an easy explanation. But it could be more complicated than that. Or in different patients, you have a different explanation, right? What interests us very much is the parosmia. It comes back to sense of smell in a very bad way. Particularly, you have parosmia also? Anosmia? You have parosmia as well? Yeah. Parosmia is typically with meat and coffee. It really smells badly. And now that I'm speculating a little bit, the vaccinations don't protect against loss of sense of smell. That's not really published. It protects you from dying, in most cases, ending up in intensive care, ending up in hospital, but it doesn't protect you really against the loss of sense of smell. So I personally, even though I've had 5 vaccines already, I don't want to get COVID because I don't want to lose my sense of smell. It is really very, very tricky. Very, very bad for the quality of life, right? Not to be able to smell. So that we're not finished yet with COVID and with Omicron and so on. No one should lose their sense of smell. It happens also in vaccinated. Yeah. You on the microphone? No. Have it for the streaming. I will repeat it. Yeah. So for some viruses, but they may printing. So you have like genetic modifications that will modify the environment for long term. So you can maybe make this happen where you don't have a new infection that they make themselves. Which virus will do that? RSV. Yeah, so the question was some viruses like RSV, respiratory syncytial virus, have epigenetic effects on neighboring cells. It's also. There's one paper on bioarchive that claims the same for SARS-CoV-2. You really have to study that histologically at the single cell level. Spatial context is important. This cell located next to that one, and most of these epigenetic techniques, as far as I know, are still dominant material, right? Yeah. That has to come down to single cell analysis. That's why for us, this is always going to be histology microscopy. I don't want to do bulk RNA-seq, for sure not single cell RNA-seq, that's not useful here. You have to have the spatial context, for instance, to study what you suggest. Question?
Thank you. These ill cells also express anesthetic receptors for LR2 or mechanism. And there are some studies indicating that polygon this receptor also anesthetic found this in your study? Mechanism, LR2, the family of FDR.
No, I don't know. It's just a bacterial cells, not glial cells. Yeah. So in human, yeah, there's very little literature on human sustainable cells. I want to know that reference, so please give it to me. I mean, these are mysterious cells that not many people have worked on ever in human, right? Some in mouse and rats, but I want to see human stuff. You know, I spend my whole life studying mice, magus now, but we have to study human to understand human disease initially, right? But please let me know that paper. Yeah. I have found no paper, 0 papers in the literature about physiology, pharmacology of human sustainable cells. 0. No one has ever studied that. Difficult to get the tissue also. Right?
So this is a kind of glial cells.
Usually people compare it to that. Yeah.
PLP was a big question.
No. I mean, and supporting cells, sustainable cells, and supporting the neurons. You can think of them as glial because it's specialized cells in the epithelium.
So I'll go down the mic. So based on your studies, do you have any hints why the incidence may be higher in people? And also whether the people study might also suggest why there is a different incidence in people versus
females. No, I don't think the GWAS study with 23andMe reported that they have 12,000 cases. It's quite a lot. But it's also based on self-reporting. You know, I must say. So to answer your question, there's a big discrepancy between subjective reporting and objective evaluation of the sense of smell, both ways, right? And so the studies we will do in the future with living patients, we will first do an objective smell taste test and then look at the tissues, not just believe what they say, right? But there's no explanation why it's more in females than in males. I was thinking maybe they go more often to a doctor to complain about it. They will, you know, could be a bias also there. There is no big studies done, as far as I know, where they take 10,000 random people, right? That's the way you should do it, and say, have you had a COVID infection? Have you had a COVID symptom? That's never been done. It's always based on clinics where people come to a doctor and complain, and it's just a very selected group of people. But it looks to be, yes, more females than males. And by the way, it's not so much in Asia. The Chinese never reported that, right? The loss of sense of smell, there was nothing. It was discovered in Iran first in March 2020, and then in Italy, first papers are from there. And I really think it's true that in Asian population, Asian ethnicity, there is less function lost in COVID, at least with the original Wuhan strain. Right? Yeah. So it worked okay.